All right, guys, so last time we just kind of introed the immune system. We're going to spend a couple of weeks for sure, maybe more, dealing with it. So let's first of all start back where we left off. And then we'll get into some of the more interesting stuff today, too. We know what the role of the immune system is, to protect you from diseases, from germs, from pathogens. But what we talked about last time was the part of your immune system that just protects you from general stuff. Not specific, just general things. So we call that part of your immune system either the non-specific, or sometimes it's called innate. And the word innate is important. What does it mean? Built in. You came with it. You were born with it. You didn't have to get it. You were born with it. So some parts of the innate or non-specific immune system that you ought to know. Number one, the most important part's the skin. It's like a barrier. It's like a wall. But it's not just a wall. It makes stuff also that helps to kill germs. In addition to blocking out germs, it kills them. Some of the secretions of the skin are acidic. Some are antibacterial. We talked about urea before. When does your body make this stuff called urea? When do you make it? No? You make it after you eat what food? When do you make urea? Yeah, well, after you eat proteins. And remember, one of the groups in the protein is that NH2 group. What's an NH2? What's it called, an NH2 group? Functional group, NH2. Silence. Everybody knows what an NH2 is? How about an amino group? When you digest proteins, those amino groups get pulled off and they combine with another hydrogen. What do you got if you put an NH2 with an H? Well, yeah, keep going. What is that? That's ammonia. Your body makes ammonia every time you eat and digest proteins. Ammonia is really, 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 really toxic. Your body, in an effort to not kill itself, the liver of your body takes that ammonia and converts it into something that's a little less toxic, and that's urea. Eventually, what organ will help you get that out of your body? What organ helps you get rid of urea? What organ? Your kidneys. And you urinate it out, right? That's why urine is urine. But also, some of that urea comes out in your sweat. And that's actually a good thing because it's toxic. And it kills things that are attempting to grow on your skin. It's poisonous. Another thing that we spent quite a bit of time talking about in the non-specific immune system was inflammation. And we mentioned that anytime tissues are damaged, whether it's cut, burn, anything like that, that there are cells in those damaged areas called mast cells, M-A-S-T, that essentially send out like SOS signals. Hey, I'm damaged. And those signals are mostly communicating with what part of your body? These SOS signals, what are they saying? Send help to. What part of your body are they saying, please help? Blood vessels. Blood vessels near the area. And what are they telling those blood vessels to do? Dilate, get bigger, and send more blood to that damaged area. It's like it's all red, puffy. And at first, like we said last time, that kind of sounds like a bad idea at first, sending more blood to a place that's cut. But the reason you want more blood there is, number one, there's platelets in that blood to help clot it. And number two, there's white blood cells in there to kill things that got in that weren't supposed to get in. The main SOS signal that your tissues release when they're damaged is histamine. There's also one called the prostaglandin, but the histamines are the ones you need to know about. They're signals released by damaged tissues that cause more blood to come to that area. Something else we talked about was interferon. And we said it's a protein that's in your blood. And what does it interfere with? Yeah, what's the virus life cycle called? The lytic cycle. It helps to stop or at least slow down virus replication. And that's something you naturally make. We talked about stomach acid. That most things that go in through your mouth, and even a lot of things that go in through your nose, end up in your stomach. And that acid, since most things are made of proteins, that acid kills most of it, denatures their proteins. So that protects you from all kinds of stuff. We also spent some time talking about your normal flora. We said that a lot of times when you use the word flora, what other word often goes with it? Fauna. And we use those to mean flora and fauna mean what? Plants and animals that live in a place. Here we're not talking about plants. We're talking about what? Bacteria. These are the bacteria that live in your body and actually are good for you. And there's so many of these bacteria in some places in your body that essentially I compared them to what? Like an offensive line in football. They're blocking for you. And they're keeping other things that might want to grow there from growing there. Simply by crowding them out. And that's kind of important. We talked about saliva and also tears. How that, you don't get many infections in your mouth, even though it's open. Some of you it's open a lot, right? You don't get that many infections in your eyes, even though they're open almost all the time. And that's because the tears, the saliva, have these proteins in them called lysozymes. What do we know lyso means? Cut, tear, break, split, destroy. And that they destroy germs, bacteria, viruses, and so on. We also spent some time talking about your mucous membranes. And we said that these are tissues that line essentially every opening into your body. And one of their main jobs is to what? What does your mucous membranes do for those openings? Filter stuff coming in. Catch stuff that's in the air. We also mentioned that if you're living in, say, Alaska, what else do your mucous membranes do for you that's kind of important? They warm the air that's coming in. And if you're living in, say, Arizona, what else do they do that's kind of important? They humidify it so you don't dry out your lungs as you're breathing. So they're important for a lot of reasons. And then the last one of these non-specific things we'll mention are pyrogens. These are proteins that make you get a fever. We mentioned that having a fever is actually a good thing in terms of helping you to fight off infection. Your immune system works better. You're able to kill bacteria faster when you've got a little bit of a fever. And if you're sick and you take Advil or Tylenol or something like that, you might feel better, but what are you actually probably doing to yourself? You're increasing the length of your sickness because you're slowing down your immune system's own effort to kill the germs. So you're probably prolonging the infection. All those things we just talked about are part of the non-specific or innate part of your immune system. Really important. It'd be hard for you to live without it, but it's not the only part of your immune system. What we're going to focus on today is the specific, or sometimes it's called acquired immune system. And it's specific because it's going after very specific things, right? It's going after specific bacteria, specific viruses. And it's acquired because you have to what? You have to get it. You're not born with it. You have to build it up over time. Some key words that we've talked about before, but we need to make sure you know before we get too deep into this. That word antigen. That's a huge word when we're talking about the immune system. What's an antigen? I talked about this a little in class the other day. What's an antigen? Yeah, sort of. I want you to think of it as any protein that's in your body that's not supposed to be in your body. We're going to call it a foreign protein. And an antigen is a foreign protein. So it could be part of a virus. It could be a bacterial protein. It could be a fungal protein. Any protein that's foreign to your body. You got a kidney transplant. It could be proteins in that kidney. They're not supposed to be in your body. So you start attacking it. Sometimes you may, instead of here seeing the word antigen, you may see antigenic determinant. These two terms are slightly different. We're going to use them to mean the same thing, essentially. Foreign proteins. If you see either one of those, proteins that are in your body that aren't supposed to be there. In response to antigens, what does your body make? Antibodies. So those two words I want you to always link together. An antigen is a foreign protein, and an antibody, it's also a protein, by the way, an antibody is, but it's a protein that you make to destroy that antigen. So antibodies help you destroy and get rid of antigens. And they're really specific. For example, let's say how many of you got a flu shot already this year? Anybody? You should go get a flu shot, by the way. You get a flu shot. What's in that stuff they inject into you? There's nothing alive in there. They're inactivated viruses. They're putting in pieces of viruses into your body, antigens. But the viruses are not working. Flu shot should not make you get the flu because it's been inactivated. It's just pieces of the viruses. They inject those into you. Whenever you have a foreign protein, what do you make? Antibodies. But those antibodies that you make are specific only to that particular flu, right? That particular thing. How many kinds of the flu are there? Thousands at least. How many are in the flu shot? 3. Okay? 3. So, how does that work? How if there are thousands of different flu viruses, how are the people encouraging you to get the flu shot saying it's worth your while if they only put 3 in there? They've been studying all year which flu viruses seem to be headed our way. There are people who that's their job, to track the flu and see which flu viruses are coming, which flu viruses seem to be coming our way. Those are the ones they put in the vaccine. Some years they guess right. And if you got a flu shot, you're protected. Other years, like last year, they guessed completely wrong. And the flu vaccine essentially did nothing for you last year. And a lot of people who got it got the flu. But point being, this is specific stuff. If you got a flu shot, you made antibodies against a particular kind of flu virus. If that flu virus gets into you, you're protected. But if a slightly different flu virus gets into you, you're probably not. That's just the way the system works. So we're going to go deep into how this works. But for right now, antigens are foreign proteins. In response to an antigen being in our body, we make antibodies, which help us to get rid of those antigens. Think of an antibody almost like a smart bomb. You guys know what I'm talking about with smart bombs? If you ever watch like I remember when we first went into Kuwait in the Iraq War, the first one. Technology was new, but they had cameras on these missiles, and they actually would show on TV. They'd say, okay, they're going to shoot this building, and it's going to go in through this window, and you could watch it on the camera on the missile. It would go straight into the window that they told you that was going into, and the building would blow up. Right? Smart bomb. That's what I want you to think of an antibody like. Because it's only targeting this antigen, the antigen that it was made in response to. All right, now we're going to talk about 2 different parts of the specific immune system. And the only one we're going to concentrate on today is the one that's on the left side of this diagram. And it's called humoral or antibody-mediated immunity. We talked a little bit about this in class the other day when we were talking about cell signaling, so this is going to tie in with what we did in class. Before we get into the whats of this, though, important word. In science, not so much today, but back in like the old days of like Greek science and stuff, what were the humors? Humors, H-U-M-O-R-S. Anybody know what humors are? They're fluids in your body fluids. So we're going to talk about what's called the humoral immune response or antibody-mediated immune response. We can use either one of those terms. But I want you to understand what's called humoral. Antibodies are big, bulky proteins, which means they can't get where? They're big old proteins. Where can they not get? Into cells. So if you have viruses inside your cells, are antibodies going to be able to help? Nope. They can only get those viruses when they're between the cells in the fluids. That's why this is called humoral immunity. These antibodies can only help you with things that are outside your cells, extracellular, in the fluids. All right, so let's talk about how this might work. And again, we're only concentrating right now on the left-hand side of this diagram, the blue part. So antigens in you. Let's say that you got the flu. Flu virus comes into your body. A couple things might happen. We're gonna go down the middle first. Most of the time, what happens first is a big cell called a macrophage, which we've talked about many times, will engulf. It'll phagocytize that virus. What have we said macrophage means? Big eater, right? Big eater. So it's a white blood cell that engulfs the virus. What happens after it engulfs it? What happens to things after they get phagocytized? They get digested. What's the word for the kind of digestion? Intracellular. What organelle makes it happen? Lysosomes. And then, let's go to another picture here for a second. We'll come back to this one. So let's say this purple cell is our macrophage. And this little sunshine looking thing, that's our virus. So right now it's getting phagocytized. It's going to get put into a little vacuole, a little vesicle, and it's going to get broken into pieces. Well, let's say these little orange triangles are pieces of a virus that's already been broken down. We might call those triangles what, if they're pieces of the virus? What word did I say you got to understand the use of them? Not antibodies, antigens. They're foreign proteins. Proteins that are in you that are not supposed to be in you. So that's what these little triangles are. This green thing, we're going to call an MHC class 2. That's this thing. It's a protein. It's part of your white blood cell. It's part of your phagocytic cell. What it's going to do, after you've taken in the virus and you've broken it into pieces, it's going to grab a piece of that virus, an antigen, and it's going to hold it up on the outside of its own cell membrane. This is another thing we talked about in class the other day. What do we call that? When a cell kills a germ and then holds up pieces of it out on the outside of its own cell membrane. What did we call that in class the other day? We called it antigen what? Antigen presentation. That's happening right here. Antigen presentation. And what's it presenting the antigen to? What does this macrophage present? What's this cell down here on the bottom? It's called a helper T cell. And essentially, what's the macrophage trying to tell the helper T cell? This thing is in us. I killed this one. I'm a bad dude. But there are lots more of them, and I need you to do what? Helper T cells don't actually do killing. But what's their job? Tell other parts of the immune system to help me out. And we need to go after this specific thing. So, what did I compare what's going on right here to? I call it going Helen Keller on it, right? That You know, through this number. Because what does the helper T cell need to know? Shape. It needs to know the shape, right? It needs to know the shape. It's learning the shape and the structure of this particular antigen. That's antigen presentation. Once it gets that shape, what's it going to go do? What's the helper T cell do once it figures out the shape and it gets activated? I compared it to what historical figure, the helper T. It's like Paul Revere. His job is to screen. Not that the germs are coming, but that the germs are already here. And here's what they look like. Go get them. Go kill them. So let's go back to the previous diagram. And let's see where we're at. So the antigen has been engulfed. The macrophage ate it, digested it, presented the antigens, and that stimulated helper T cells. Those helper T cells then are going to start sending out the message. And the main thing that they're going to send messages to in our case, on the left-hand side of this diagram, are these cells called B cells. Or you may hear them called B lymphocytes. A lymphocyte's a fancy word for what? White blood cell. It's a specific type, B cell, specific type of white blood cell. It turns out, though, that these helper T cells only stimulate B cells that are capable of dealing with what? This is quite specific. They only stimulate B cells that are capable of dealing with that specific antigen. You've got lots of different B cells. Some of them can't help you against that. Some of them can. So the helper T cell stimulates only the B cells that are capable of killing that antigen. And what's the first message it's telling those B cells? What's happening as we go down that diagram, you think? You've got lots of different B cells, but do you have a bunch of each one? No. So what's the first thing that needs to happen? The one that's specific to that antigen, the helper T cell is telling it to clone itself, to make bunches and bunches of copies of yourself. So that happens first. These B cells start to clone themselves. Thousands and thousands of copies of the B cells that are specific to that antigen are made. Most of those B cells will go through almost like a state of metamorphosis. And they'll turn into these big cells called plasma cells. So the cloned B cells, most of them become plasma cells. And they're called plasma cells because they have a lot of rough ER. And what do we know cells that have a lot of rough ER? What are they doing? Making proteins and what? Secreting them. What kind of proteins do we need to be making and secreting if we're at plasma cell? Antibodies. And they make hundreds of thousands of antibodies per second. Antibodies that are specific only to what? That particular thing, that particular antigen. And these antibodies go into your fluids, your body's fluids. And they float around. And let's use an analogy here. Let's say that you guys are all floating around, and our room is a body, and you guys are all floating around in there. And these little markers are antibodies. And they're only made against a specific type of antigen. So Nathan, he's one of those antigens. So these antibodies are floating around, and my antibody sticks to Nathan. Grab onto it there, Nathan. And Jordan here, he's another antigen. Antibody sticks to him. It doesn't stick to any of the rest of you. What does that antibody do to these two guys? it may not kill directly. Now, one thing it might cause to happen is it may cause all these germs to kind of clump together, or it may be like a beacon. You've got in your body these macrophages that eat things, but they're kind of big, and think of them as that big and dumb and slow. They're not going to be out looking for Nathans in your body. But if there's a big flashing light on Nathan saying, "Here I am," what will the macrophage do? It'll go get it. And that's kind of what the antibody does. You might think of it like a mark for death. Okay? So Nathan is now marked for death, and so is Jordan, and the macrophages will go kill them. So what I'm saying is antibodies may not kill, but eventually they cause them to be killed. Another thing that might happen, a lot of times when you go to the doctor and you have a virus, what does the doctor tell you in terms of advice he's giving you when you have a virus? Again? Yeah, well, man up, tough it out. What else? Treat the symptoms and drink a lot of fluids. Why would he tell you to drink a lot of fluids? Let's say Jordan here's a virus. I've already marked him. But I need to get him out of my body. I'm a white blood cell. I don't want to touch Jordan because he's dangerous. I mean, look at him. He's a dangerous guy, right? So, but he's got a mark on him. As a white blood cell, if I touch him, maybe I get infected too. So I don't want to do that. So what might I use that antibody as? I'm going to use it like a handle. And I'm going to grab that antibody, and then I'm going to escort Jordan to where? Where am I escorting him to? The kidneys. And what are the kidneys going to do? They're going to filter him out of my body fluids, and then I'm going to flush him, right? Okay, that's essentially why you need to drink a lot of fluids, because one of the ways you get rid of these germs is that you urinate them out. And then one of the ways that antibodies help you is they act almost like little handles that attach to those germs. Your white blood cells can grab those handles, escort them to the kidney, where you filter them out and flush them essentially. So you need to drink a lot of fluids to do that. All right, so almost all these B cells that got cloned turned into plasma cells. Almost all of them. A few of them didn't. A few of them hang around as what we call memory cells. What do they remember how to do? What kind of memories do memory cells have? They know what the germ looks like, and they know how to make antibodies against it. Here's the thing. This process that I just described to you, from macrophage eating antibody or eating antigen to making antibodies to getting well, how long does that normally take? How many days does it take from exposure to your well most of the time with viruses? It can take, anywhere from a week to 2 weeks. You're going to be sick that whole time. But if this thing's already been in you, and you've already successfully fought it off, if it gets in you the second time, what happens? The same exact germ gets in you a second time. You already know how to fight it. You already remember how to fight it, right? So all these initial steps get skipped, and you start here, essentially. And you make so many antibodies so fast.
Mr. Dunn, Mr. Hawley, Mr. Campbell, Ms. Clark, Ms. Allen, please come to the guidance office. Mr. Dunn, Mr. Hawley, Mr. Campbell, Ms. Clark, Ms. Allen, please come to the guidance office. Thank you. Bye-bye.
Okay, they're in trouble. Regardless, the memory cells, what they allow you to do is skip a bunch of these initial steps. So instead of taking 4 or 5 days for your immune system to get rolling, it gets rolling immediately. So you make lots of antibodies really, really fast. So many, so fast that what doesn't happen the second, the third, the fourth, the fifth exposure. Yeah, and what doesn't happen to you? What does not happen to you? You don't ever get sick. You don't ever show any symptoms, even though this thing's in you. So that's the beauty of memory cells. Once you fought it off, you're probably not going to get it again. Who in here has the most memory cells, probably? like me. I've been around a lot more years than you guys. Fought a lot more battles, won those battles, and know how to win them, right? And that will, eventually you fought so many battles you start falling apart, right? Hopefully I'm not there yet. But the more memory cells you have, the less likely you are to get sick. But the key thing to remember is those memory cells only have a memory of what? That one thing. So that you're building up more and more memory cells across your life. Vaccinations help you because they cause you to make what? Memory cells and antibodies. So the memory cell. Here's the thing. This is something I didn't talk about. These plasma cells. Most of the B cells became plasma cells. What happens to those plasma cells after just a handful of days? They commit suicide in a process called apoptosis. And that's an important word to know, by the way. Apoptosis. It's called programmed cell death. They die in just a few days. They commit suicide. Which means what happens to the amount of antibodies in your blood against this particular thing? After you've gotten well, do you need those antibodies floating around in your blood? No, they go away. So almost all the antibodies are gone. But the memory cells hang around hopefully for your whole life. They're there. And if that thing ever shows up again, you can respond to it really, 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 really fast. Does that make sense? So that's called humoral immunity. It's called that because antibodies can only work where? In the fluids. Can't get into the cells. Only work in the liquids. And it's antibody mediated. Essentially meaning that you're making antibodies in response to specific what? Antigens. Right. This can also get kicked into a gear instead of a macrophage displaying the antigens. Occasionally, just antigens floating around in your body fluids, what we call them free antigens here, can bind to B cells and activate them. So it's usually going to get activated this way, but it might also get activated that way, with just antigens floating around in the fluids, causing B cells to become active. One thing we mentioned in here before, HIV, AIDS. Why is it so damaging to you? It wipes out Paul Revere. Okay? It wipes out the helper T cells. Which means not only does the antibody mediator response usually not get kicked into gear, turns out the helper T cells are also important on the other side of this too. So what I'm saying is by HIV killing your helper T cells, it essentially wipes out what? Your whole acquired immune system. Which essentially means you're left with what? Skin and stomach acid and stuff like that. That's pretty much all you got in terms of an immune system. It's not very effective. So we will briefly mention this other side today. I'm not going to go too deep into it. The other side of this picture is called the cell-mediated immune response. And it's important to help you get rid of germs that are where? In cells. So this would be particularly important for what kind of infections? Viral, because they get in the cells, right? And the antibodies can't get there. Before we get into how this works, I want to back up to a picture, let's talk about this picture again for a second. All right, so now we're going to look at this side of this diagram. And this orange cell is just a normal cell in your body. It's not a white blood cell at all. What's the word for a normal cell in your body? It's a somatic cell. And it turns out that somatic cells, like the macrophages, have a protein, an MHC, that they can use to present antigens. This one's called an MHC class 1 protein. All the cells in your body have that. And typically, what kinds of proteins are they sticking up on the outside of their cells using that MHC? What do those little blue triangles represent? Not necessarily antigens, but proteins that are in that cell. And normally, what kinds of proteins should they be? What kinds of proteins should your normal cells be holding up? Normal proteins. Just proteins that are inside them. Normal proteins. So you have different kinds of cells. Helper T cells are still involved, but you also have a type of cell called a cytotoxic T cell. Or I'm going to call it, in this case, a killer T cell. Same thing. They're coming around and checking these little proteins out. If it's a normal protein, what does that tell the helper T cell and the killer T cell? This cell's good to go. Don't mess with it. Leave it alone. It's healthy. But what might cause a normal cell to hold up some proteins, to present some proteins that aren't normal? What might cause weird proteins to be inside that cell? The virus, or maybe what else? What else might cause a cell to hold up, to present weird proteins? Maybe like cancer in patients. Either way, this thing is now presenting strange, abnormal proteins. What's that cell essentially telling the immune system? What's the message that cell is trying to communicate to the immune system? Something's in me that's wrong. You need to kill me before what? Before I cause everybody else to die. So it's like almost like a suicide note that it's holding up. It's not going to commit suicide. It's going to ask to be killed. Okay? And what will end up happening is that eventually the killer T cell or the cytotoxic T cell will recognize that antigen, and it will come over, and I talked about this in class the other day too, that it'll essentially like go Wolverine on it. What I mean by Wolverine is it's going to poke holes in it. It's going to use this protein called perforin. What does perforate mean? Perforate something, what do you do to it? Poke holes in it. So this protein perforin will stick holes in that cell, killing it. And the internal parts of that cell are now going to leak out. If there are viruses in there, now what can get to them? If they leaked out, what can get to those viruses now? The antibodies. They can't get there otherwise, right? Because it's inside. So what I'm telling you is that the cell-mediated stuff, the stuff that we just mentioned, and the antibody-mediated stuff, they're happening at the same time. And they're working together to help you get rid of very, very specific kinds of germs. And I think that is a good place for us to stop today.